Chapter 6, Culturing Microorganisms. The learning objectives for this topic are: describe methods of collecting clinical specimens from the skin and from the respiratory, reproductive, and urinary tracts. Describe the two most common methods by which microorganisms can be isolated for culture. Describe 6 types of general culture media available for bacterial culture. Contrast refrigeration, deep freezing, and lyophilization as methods of preserving cultures of microbes. The overview for this topic are: we're going to discuss clinical sampling, obtaining pure cultures, culture media, special culture techniques, and preserving cultures. First, let's talk about clinical sampling. The diagnosis and treatment of disease often depends upon isolation and correct identification of the pathogen that is causing the disease. So these samples that are taken from patients who are sick need to be handled with universal precautions. Well, what do I mean by universal precautions? They are handled such that the sample is not contaminated with organisms that were not present in the patient. So we want to keep organisms out. And we want to prevent contamination of the surroundings. You don't want pathogenic bacteria all over your lab or anywhere else. And then we use transport media to prevent a change in the sample. You don't want an organism that could cause disease in high numbers to go from low numbers in a sample to going to high numbers. On the other side, you don't want very delicate pathogenic organisms dying on the way to the lab, thereby indicating that they're not present in the sample. So transport media controls pH, presence or absence of oxygen. You also want to control the temperature. Generally we want it cold. When we talk about preserving samples, we'll talk about refrigeration. And speed. Generally speaking, clinical samples need to get from where they're being sampled to where they're going to be cultured in a very short period of time. Now, for the objective that talks about how to collect clinical samples, there is a lovely table in Chapter 6, Table 2, which will go over that for you. Let's talk about how we get pure cultures, how we get cultures that have just one organism. With the clinical samples, generally speaking, up until recently, diseases were caused by just one organism, the pathogen, and we wanted to get a pure culture of it. So here are some terms. Inoculum. That is what we're putting into the media to grow. So anytime we're putting an organism or a clinical sample in, that's an inoculum. So it can be environmental, clinical, which has been the focus of microbiology up until recently. And then there's lab cultures, pure cultures that we've been growing in the lab for long periods, and they're kind of like our pets, versus clinical environmental, those are wild cultures. Medium. Now I use the term medium without defining it. Let's define it now. Medium is the food that we feed the organism, that we feed the inoculum. Now it can come as a liquid or a broth, or it can be a solid, an agar. Now this would be an example of an agar slant. You've also worked with agar plates. There's benefits to either one. With the liquid, it's harder to get a pure sample. You have to do dilutions. With a solid agar, you can spread them out, stick the bacteria on the surface of the plate, and you get individual colonies. You can take that colony, take it to the broth, and grow it in pure culture faster in the broth than you would on the agar. So generally speaking, when we're obtaining pure cultures, we go with the solid, then we maintain them in the liquid. Let's talk about how we use the different types of media to do the actual obtaining of pure cultures. So you can take your clinical sample, your inocula, and you can do a streak plate. So what we're doing is we're taking a loop full or a swab, and we put it on part of the plate. Now you can tell that there's all of the microbial growth is running into each other. This is what we want. We want individual colonies that hopefully arose from just one organism. See, we've got 2 different kinds here. So we sterilize our loop, and we go through our initial streak and we spread it out some more. Now we're starting to get some individual colonies here, not good enough. So we sterilize our inoculating loop again and we come through, and you can see we've gotten few enough numbers that we can get individual colonies, and we would take that and we would streak it again to make sure that it's a pure culture. We would stain it and hopefully do some testing on it. So we call that a streak plate. Now there's another method where we can use both broth media and solid media. We take the initial sample, of which we know there's way too many to be able to get individual colonies. So what we do is we do sequential inoculations into sterile broth at known volumes. So we take the initial sample, we take 1 ml of it, put it into 9 mL. We call that a 1 to 10 dilution. We do that again, we got another 1 to 10 dilution, and then we take that and we spread it on the surface of the plate. So you can see here we have 10 times as many colonies as we do here. We would call these spread plates. Now this figure is actually an example of a series of pour plates. That's where we take the broth, mix it into the agar while it's still liquid, but cool enough that you don't fry your poor little guys, and then you pour it into the agar plates. See, we have subsurface colonies growing inside the agar. With spread plates, of course, you would only have them growing on the surface. Let's go over the different types of organisms and the types of media that they need. So most of the organisms that you're going to be dealing with in lab are heterotrophic. What does that mean? They're getting their carbon molecules from other organisms. In other words, they are not autotrophic, they are not photosynthetic. Most of them we can grow in standard media such as tryptase soy broth or nutrient broth. Now, tryptase soy broth and nutrient broth, or tryptase soy agar or nutrient agar, are both examples of undefined culture media. In other words, we're putting in soy. We don't know exactly what the chemical composition of the particular soy plant was that went into the broth. It depends upon where it was grown and how much fertilizer it got, etc., etc. Now, we also have some standard media that is defined. We know exactly what's in it. We use molecules like purified glucose, we use purified salts, and we can grow some heterotrophic organisms using that. Now, some heterotrophic organisms are fastidious. Fastidious is a fancy-dancy term for picky. They're very picky. They just don't like these standard media, and if they don't like it, they don't grow. Actually, what's happening is they need more growth factors. They need things like cofactors, they need vitamins, they need amino acids that they can't produce themselves. So they're really not all that picky. They're just not being provided with what they need. So generally when we grow fastidious organisms, we grow them in undefined media unless we know exactly what they need, and then we can grow them in defined media. Now, obligate intercellular parasites are the ultimate in fastidious organisms. We cannot grow them in sterile culture media. We can't grow them in dead stuff. We have to grow them in either their host organism. I have animal here, but some of them you have to grow in plants. Or you have to grow them in live cells. We'll be talking more about that. Then there are some organisms that it doesn't matter what we provide them, we cannot grow them. We call these unculturable organisms. So to study them, we have to use molecular biology, and oftentimes we're studying them in biofilms or in non-pure cultures because they're getting their growth factors and other things from their buddies in the biofilm. So let's talk about a particular type of culture media called selective media. Selective media favors the growth of the wanted organism, or they inhibit the growth of the unwanted organism. So in other words, we're taking a mixed culture and we're selecting for the organism that we want, or we're selecting against the one that we don't want. Now, some of the things that can inhibit the growth of unwanted organisms are stains. Stains eventually kill everything, but some organisms are more resistant to the stains. So a lot of our selective media we will put stains in. So with these stains, They all inhibit gram-positive bacteria without affecting the growth of the gram-positive or the gram-negative bacteria. Now, Sauerbrod dextrose agar is an agar that has high concentrations of sugars and also a low pH. And this selects for fungi and inhibits bacteria. So if you're trying to grow fungi, oftentimes we'll use selective media for fungi because they grow slower than bacteria. And if you just put them on standard media, the bacteria will outgrow the fungi.